We've been talking about what Francis Crick conceived of, which is genetics, the information transfer from DNA to RNA to protein. But what he couldn't conceive of is that there are other forms of information transfer. There are other ways of modifying that information. We don't call that genetics, we call that epigenetics. So next, let's look at the process of epigenetics and take a couple of examples of epigenetic modification of genes. To understand epigenetics, we first have to look at the structure of chromosomes. We've got a DNA double helix. That's at the top of this diagram. We wrap that DNA around a protein called a histone. Now, DNA is negatively charged. DNA is an acid, and acids have negative charges all over them. And we got 3 billion base pairs, so 3 billion negatively charged atoms there that we can't put together because negative charges repel each other. So the first thing we've got to do is we've got to screen off those negative charges with positive charges. We can find positive charges in certain amino acids that are put into a protein, and that protein is called a histone. And those positively charged proteins called histones are used to screen off the negative charges. We wrap the DNA then around the histones to neutralize the charge on them so that we can pack these molecules together very tightly. That wrapping is called a nucleosome. So when we wrap DNA around a histone protein several different times, that's called a nucleosome. And then the nucleosomes are condensed into chromatin, and the chromatin is what makes up the chromosome, this familiar looking structure that we've probably seen in other biology classes. I said the information flow from DNA to RNA to protein is genetics, but what about other kinds of information flow? How do you do that? Well, you could modify whether a gene can be turned on or off. You can modify whether RNA polymerase can get to it easily. You can modify the structure of the DNA itself, the chemical structure of the DNA, and we're going to see examples of both of those in the next couple of slides. So one kind of epigenetic regulation is acting on the histone proteins and making the histone proteins better or worse at wrapping DNA. If the DNA is in a more open structure, then the RNA polymerase can get in, and that's what we see here. If the DNA is in a more closed structure, then the RNA polymerase can't get in to do its job, and that's what we see here. So by adding an acetyl group, what's called acetylation of the histone proteins, we can open up the structure and tend to turn genes on that are located within this region of the DNA. If we methylate the histone proteins, if we add a methyl group to those histone proteins, then we tend to pack the histones more tightly next to each other, the nucleosomes more tightly next to each other. And because we don't have any openness in the DNA there, then the RNA polymerase can't get in and transcribe that area, and so that gene is effectively turned off. Another way that we can modify DNA is by directly modifying DNA with a chemical grouping called a methyl group, CH3. And so when we add methyl groups onto cytosines, then that doesn't interfere with the base pairing properties of cytosine, but it does interfere with the ability of RNA polymerase to form a transcript from this segment of DNA. And so this, again, effectively turns off the gene at that point. This causes the gene to not be transcribed, and if it's not transcribed, then it can't be turned into a protein. And so we can block protein synthesis from that gene in this way and turn that gene off. So if the DNA is unmethylated, then we call that state on. If the DNA is methylated, we call that state off. This is another epigenetic tool that we can use for modifying the structure of DNA and thereby modifying whether that DNA can be transcribed into messenger RNA.